Hi everyone, this is Jonathan Cedillo, student at Grand Canyon University, and today I'm presenting to you the effect of nanoparticles in the pulmonary surfactant. Presentation for the class Biology 520 consists of Human Physiology 2, Dr. Joan Tassoni. Research in pulmonary drug delivery has become a novelty nowadays, but there are some challenges at the molecular level that block the passage of pulmonary drug therapy. To surpass these challenges, inhaled causative disease factors are being studied. In the article named Physiochemical Properties of Nanoparticles Affecting Their Fate and the Physiological Function of Pulmonary Surfactants, key player substances are studied. Interestingly, when substances are introduced in a patient, such as inhaled nanoparticles, they target the pulmonary surfactant. In addition, the surfactant protein B and surfactant protein C are critical in the correct activity of the pulmonary surfactant, while the surfactant proteins A and D are crucial in the correct autoimmune activity of the lungs. Scholar claims that the pulmonary surfactant is an important target that will allow an efficient delivery of drug therapy. However, maintaining the structure of physiology of the surfactant is important due to its correlation to the low alveolar surface tension and prevent the lungs from collapsing, because it can prevent airborne disease such as asthma, cystic fibrosis, or COVID-19. Continuing, the physiochemical parameters of nanoparticles such as size, hydrophobicity, hydrophilicity, charge, and shape are a challenge to the application of drug therapy. The hydrophobic surfactants proteins B and C are important for the integrity of the pulmonary surfactant film. After inhaled nanoparticles have been applied to study their behavior, authors of this paper claim that the coronal structure could be formed. Examining the physiochemical characteristics of nanoparticles and their effect in the pulmonary agents are getting the attention of scholars. In this paper, techniques on how to analyze the factor in the pulmonary surfactant are presented due to the important activity of the physiochemical properties over the hydrophobic proteinic absorption. Background: the pulmonary surfactant is principally made of lipids and proteins that is spread across the epithelial cells of the respiratory system. Lipids constitute 90% of the pulmonary surfactant, having phosphatidylcholine as contender number one, since it is the phospholipid that is the most present. And fully saturated dipalmityl phosphatidylcholine is roughly 41%. Other phospholipids such as phosphatidylinositol, phosphatidylethanolamine, phosphatidylglycerol, and cholesterol are also present in the surfactant. Additionally, the film is also composed of only 8% of hydrophilic and hydrophobic proteins. Furthermore, according to scholars, the main physiological function of the pulmonary surfactant is the maintenance of the low surface tension at the air-liquid interface, preventing the lungs from collapse. Between the several characteristics in the pulmonary surfactant, the surfactant proteins B and C are critical in its integrity. Implementation of inhaled nanoparticles that interact with the film of interest and the absorption of hydrophobic surfactant proteins B and C can potentially affect normal activity of respiration. Moreover, in this study, the effect of the physiochemical properties of nanoparticles over the hydrophobic proteins absorption and techniques to downregulate their contact with nanoparticles is studied and presented. Here I'm showing you the result of the experimentation of this article. Here in figure 1a, authors of the article are presenting the surfactant lipids and proteins and their percentage composition in epithelial cells across the respiratory system. Lipids are the main component, covering 90% phosphatidylcholine or PC, which is the most present, in which the fully saturated dipalmityl phosphatidylcholine or DPPC is about 41%. As presented in the figure, other phospholipids encompass the composition of the endothelial cells of the respiratory tract. Figure 1b shows the components of a single phospholipid. Figure 1c shows the different structures that a phospholipid can build and the structures of surfactant proteins A, B, C, and D. To determine the invasion of nanoparticles at the pulmonary surfactant film, Lagmore Drove is the technique used to analyze this event. Figure 2a1 shows the surfactant material that merges with an organic solvent which forms a layer at the surface of a liquid after solvent evaporation. Figure 2a2 shows a relative area of the lipid molecules versus surface tension isotherm of the D-palmitoyl phosphatidylcholine, analyzed with the Langmuir-Tropsch technique. When stress factors such as compression occur, the pulmonary surfactants go through different stages. In one of them, tails of phospholipids are scattered and topologically oriented towards the air phase. In addition, the pulsating bubble surfactor meter is used to analyze the activity of surfactants, which is shown in figure 2b1. When reaching equilibrium, the distance of the bubble from the center to the diameter changes between two positions. In addition, two important factors play a critical role to measure surface activity. These encompass surface tension, which is symbolized by the gamma symbol, and minimum surface tension, which is the surface tension at the least bubble radius as presented in figure 2b2. In figure 2c1 shows analysis of the technique known as constrained drop surfactometry. In this technique, a droplet can be enlarged and reduces at the rate of common inhalation and exhalation with the aid of an electronic syringe. Furthermore, surface tension can be calculated by means of mathematical approaches such as Laplace equation of capillary. Figure 2c2 presents the relative area versus surface tension. Results reveal that the surface tension reduces to approximately zero. On the other hand, the relative area is reduced by approximately 20%, suggesting a healthy film with low compressibility. During the event of the film enlargement, as expected, the surface tension rises slowly, adhering to the compression path, and the deviation of the contact angle from its theoretical and mean value was reduced, suggesting a soft and high expandability of the film. Figure 3 shows nanoparticles and their surface charge density, which authors of the paper state that these might affect the corona structure of the lipids. After using a coarse-grained molecular dynamics simulator, results reveal that nanoparticles with an approximate charge of 60 millivolts, their hydrophobicity might dictate the lipid coronal shape. As we can see in the figure, the hydrophilic side of the phospholipids tends to feel attracted to the hydrophilic side of the nanoparticle at an angle that is less than 20 degrees, forming a corona by a layer of coated lipids. This also means that the hydrophobic side of phospholipids will like to interact with the hydrophobic side of the nanoparticles, forming a greater angle between the surface of the particle and water droplets of about 70 degrees. However, this time only a monolayer was formed. Finally, in the middle of the figure, we can see that a bicycle-coated lipid was formed. Here, in figure 4, authors of the paper show the efforts after investigating the interaction with silica nanoparticles in the presence and absence of dipalmitoyl phosphatidylcholine or DPPC, coating at the epithelial cells 16-HBE and A549 adenocarcinoma cells. Results demonstrated that the 16-HBE cells had a greater concentration of silica nanoparticles than A549 cells. In addition, both cell lines downregulated the interaction of DPPC-coated silica nanoparticles shown in figures 4A and 4B. Furthermore, DPPC showed a significant downregulation in the cytotoxicity of silica nanoparticles in 16-HBE cell line. This might potentially occur due to the activity of ABC transporters regulated by DPPC. Continuing, the APC transporters process was analyzed using calcine AM assay, which revealed that in 16-HBE cells, the activity of ABC transporters was downregulated, which led to the interaction between high-level silica and restoration activity of the ABC transporter by DPPC, as shown in figure 4C. Nonetheless, the cell line A549 presents a higher ABC transportation activity than 16-HBE cells, but the activity of this cell line, ABC transporters, was not significantly affected by silica nanoparticles in the presence or absence of DPPC coating as shown in figure 4D. Figure 5 shows the results of the investigation of the cellular uptake of dipalmitoyl phosphatidylserine, or DPPS, DPPC, and PVA-coated curcumin loaded acetylated dextran nanoparticles in the A549 cell line and raw 26.4.7 macrophage. Data reveal that in contrast to the A549 cells, greater levels of nanoparticles were interacting with raw 264.7 macrophage for all nanoparticles. And DPPS Coated nanoparticles demonstrated greater levels of interaction with raw 26.4.7 and A59.4 cell line than the one from DPCC and PVA-coated curcumin as presented in Figure 5A. Figure 5b shows the effects of surfactant phospholipids using coated and uncoated single-wall carbon nanotubes. Results reveal that compared to free surfactant phospholipids, the phagocytosis levels of coated phospholipids of single-wall carbon nanotubes were downregulated. Continuing, the surfactant proteins A and D are part of the collectin family. These enhance pathogen activity and inhale particles to promote their clearance from lungs. This means that after nanoparticles interact with these proteins, macrophage activity will be initiated. A proposal of the mechanism of surfactant proteins A and D is the protein-mediated agglutination of nanoparticles. Figure C shows the results after analyzing the surfactant protein A induced calcium-dependent agglutination of polystyrene particles, which initiated macrophage activity of raw 26.4.7 cell line. However, it resulted in calcium-dependent inhibition of the polystyrene particles' self-agglutination with blockage in macrophage activity. Conclusion. Pulmonary surfactants prevent nanoparticles to cause disorders of the respiratory system. So naturally external nanoparticles might cause inhibition of homeostasis of the respiratory tract. This article presented how nanoparticles can cause physiological damages to pulmonary surfactants. Results demonstrated that hydrophobic nanoparticles absorb critical proteins for the correct activity of the pulmonary surfactant, such as surfactant proteins B and C, resulting into negative effects of the film of interest. Interestingly, this event can be inhibited by modifying hydrophobic nanoparticles, turning them into a hydrophilic compound. Overall, smaller nanoparticles with a stronger hydrophobicity and a positively charged demonstrated a high level of lipid absorption, on which hydrophobic nanoparticles have a significant attraction to absorb surfactant protein A, while hydrophilic and negative nanoparticles prefer surfactant protein D. However, the major damage of the pulmonary surfactant comes from the absorption of the surfactant proteins B and C. Moreover, the interaction between the respiratory system and nanoparticles initiates activities such as mucociliary clearance and macrophage, which try to eliminate the particles. But in some cases, this activity can be inhibited. Furthermore, scholars claim that the parameters of nanoparticles also affect absorption of pulmonary surfactants. In addition, the absorption of pulmonary surfactants is a difficult topic, and the particles do not only absorb one component of the surfactant, but its entirety. Depending on the absorption of the amount of surfactant proteins, that is the amount of damage that the pulmonary surfactant will receive. Finally, quoting the scholars responsible for this publication, the consequent effect on the fate of nanoparticles in vivo, including intrapulmonary clearance and retention, extrapulmonary metastasis and distribution, deserve more attention and further study. Thank you for watching my presentation. See you next time.